Let's compare the packaging of DNA in chromosome with a day-to-day example. In this, we can see how the scattered clothes can be folded to pack it in a suitcase in a compact manner. Similarly, the DNA is also folded and condensed to form a compact structure in the form of a chromosome. In 1974, Kornberg proposed that DNA and histones are organized into repeating subunits called nucleosomes. Chromosomes are basically composed of chromatin. Chromatin primarily consists of DNA and proteins. The proteins are of two major classes: histones, which are basic proteins having positive charge, and non-histones, which are acidic proteins having negative charge. We will see the packaging of DNA in the chromosome. Nucleosome forms the lowest level of chromosome organization. Thus, it forms the structural unit of chromatin. The nucleosome core consists of histone octamer, also known as histone core, and 146 base pair of DNA wrapped almost 1.8 times around the histone octamer to form nucleosome of size 11 into 6 nm. The histones are positively charged, having basic amino acids lysine and arginine, and the DNA is negatively charged because of the presence of phosphate group. It is believed nucleosome is stabilized as the positive charge on histones due to the exposed amino group of arginine and lysine allows it to interact with the DNA having negatively charged phosphate group. Histone core do not bind randomly to the DNA. Rather, they tend to position themselves at certain locations which appear to depend on a local abundance of AT base pairs. The DNA associated with the nucleosome core is known as core DNA. The histone core comprises the following four histone proteins: H2A, H2B, H3 and H4. In addition to the four conventional core histones, several alternate versions of H2A and H3 are also synthesized in most cells. They have specialized functions. For example, a variant of H3, Ken-Pei, is present in centromeres and is involved in kinetochore assembly. One nucleosome core is connected to another with the help of linker DNA, which is around 8 to 114 base pairs in length. The histone that is associated with the linker DNA is H1 histone and is therefore known as linker histone. It binds to the nucleosome at the entry and exit points of the DNA and stabilizes the complex. It is believed that the core DNA is nuclease resistant and the linker DNA is susceptible to nuclease attack. The histone core consists of two copies each of histones H2A, H2B, H3 and H4 assembled into an octamer. Let's see the assembly of histones in a nucleosome core. The assembly of nucleosome is initiated by the formation of H3H4 tetramer. The tetramer then binds to double-stranded DNA. The H3H4 tetramer bound to DNA recruits two copies of H2A, H2B dimer to form histone octamer. The core histones have similar structural features. They consist of a globular histone fold and N-terminal tail. The globular histone fold is composed of three alpha helical regions separated by two unstructured loops. It has a short helix alpha 1 followed by loop L1, long helix alpha 2, a second loop L2 and another short helix alpha 3. Globular histone fold domain mediates the assembly of the histones. Histone fold extensions can be of two types: C-terminal extension and N-terminal extension. The N-terminal extension is also referred to as N-terminal tail. N refers to the end of the polypeptide chain where the amino group is located. The exposed N-terminal tails are not required for the association of DNA with the histone octamer. Instead, they are subject to various post-translational modification, such as methylation, acetylation, phosphorylation, etc., which in turn influence the gene expression. The details of the histone modification will be discussed in a separate video. The tails of the histone molecules protrude from the nucleosome. It is believed that the chromatin can expand and contract in response to the chemical modifications of histone H1 and histone tails, which protrude out and are accessible to enzymes that add and remove chemical groups such as methyl and acetyl groups to undergo methylation and acetylation respectively. Let's discuss the packaging of DNA to form higher order chromatin structure. DNA molecules wrapped around histone core to form nucleosome is the lowest level of chromatin organization, in which a 2 nm DNA molecule is first condensed into 11 nm nucleosome. Nucleosome organization gets further condensed and coiled to produce a solenoid of 30 nm diameter. In the solenoid model, linker DNA gently curves as it connects consecutive core particles, which are organized into a single continuous helical array containing about 6 to 8 nucleosomes per turn. Maintenance of 30 nm fiber depends on the interaction between histone molecules of neighboring nucleosomes. Linker histones and core histone both been implicated in higher order packaging of chromatin. Core histones of adjacent nucleosomes may interact with one another by means of their long flexible tails, which in turn stabilizes the 30 nm fiber. The structure is further stabilized by the histone H1 protein. These type of interactions are thought to mediate the folding of the nucleosome filament into a thicker fiber. Two most popular models to explain the 30 nm fiber are the zigzag model and the solenoid model. In the zigzag model, linker DNA is present in a straight extended state that crisscrosses back and forth between consecutive core particles, which are organized into two separate stacks of nucleosome. But in the solenoid model, linker DNA gently curves as it connects consecutive core particles. Another difference is that linker DNA passes through the central axis of the superhelix in case of zigzag model, but in this case, linker DNA does not pass through central axis of the superhelix. In zigzag model, entry-exit points of nucleosome is accessible. In solenoid model, the entry-exit point of nucleosome is inaccessible. Additional folding of the 30 nm fiber is required to further compact the DNA. 30 nm fiber forms a series of large supercoiled loops of DNA anchored to a proteinaceous structure referred to as nucleus scaffold, mainly composed of non-histone proteins. Two classes of proteins contribute to the nucleus scaffold. Topoisomerase II, SMC protein known as structural maintenance of chromosomes. Topoisomerase II regulates the degree of supercoiling and also untangles the DNA molecule. SMC proteins, which are of two types, cohesin and condensin, in which cohesin is involved in linking together the two sister chromatids, and condensin is essential for condensation of chromosomes as cells enter mitosis. The loops of chromatin fibers are further folded and condensed into thicker fibers, resulting in more compact structure. During mitosis, chromatin undergoes maximum compaction to form metaphase chromosome. This highly condensed chromosome plays a major role in organizing and packaging the giant DNA molecule into structures that will facilitate proper segregation. Let's discuss some objective questions related to the topic. Which histone stabilizes the nucleosome complex H1? Lysine and arginine are which type of amino acids? Basic. What separates one nucleosome core from another? Linker DNA. The histone forms a structural core known as histone octamer. How many types of histones are there? 5. The model of a 30 nm fiber in which DNA is present in a straight extended state that crisscrosses back and forth between consecutive core particles, zigzag model. One solenoid model is composed of 6 nucleosomes. Which of the following is nuclease resistant? Core DNA. The protein that links sister chromatids together, cohesin. Which histone is not present in the nucleosome core? H1. Which type of histone molecule helps in the assembly of kinetochore? Kenpe. Type of histones involved in nucleosome core? 4. What is the charge in histone which allows it to bind to the DNA? Positive. Which of the following is not a characteristic of the nucleus scaffold? Topoisomerase 1. Type of histone binding to the linker DNA? H1. Which type of DNA sequence are preferred by the nucleosome for binding? AT-rich. What marks the difference between the solenoid and the zigzag model of 30 nm fibers? Linker DNA. Histone is rich in dash type of amino acid, both lysine and arginine.